some of the key things you should remember for the fungi and the protists. So the way I like to do it is kind of write small over here and leave like a little margin, maybe a 1-inch margin on this side, where you can put some categories. And then list things in the categories, like oncogenic viruses, and list them, right? So kind of try to write on like maybe 3/4 of it here and leave a little margin. So my sheet is a little bit different than yours. Let's start with the viruses. So remember, first of all, that outside of HIV, so starting with HSV 1 and 2, all the way to HSV 8, right? That's HSV or HHV or the name, right? So you should know those. Like HSV 1 and 2 is what? Herpes, cold sores, right? So it's human herpes virus 1 and 2, herpes simplex virus 1 and 2, cold sores, right? Genital herpes. So you should know the names. Then HSV3 is also HHV3, HSV3, chickenpox, and what else? There's one that's not on there. Shingles, and what else? Varicella, right? So VZV. So you should know the names. HSV4 is Epstein-Barr, which can be abbreviated EBV. HSV5 is cytomegalo. And then remember, you have to know 6 and 7, rhodiola. And then there's 8, and that's Kaposi sarcoma. Okay, so then we have our hepatitis A through E, which what's important is to remember the modes of transmission for those. And then we have influenza, so we'll get to that. We have measles, mumps, rubella, the MMR. We talked about those. HPV, polio, Ebola, and Zika. So let's start off with HIV. What do you think is important to know about HIV? All kinds of good stuff, right? So first of all, what kind of virus is it? A retrovirus. So it's a retrovirus. Now, what types of cells are targeted by HIV? What's the key defining factor that they have? CD4 positive cells. And what else? What's the co-receptor that we've talked about a lot? CCR5, chemokine receptor 5. So you need to remember that. Chemokine receptor 5. And we talked about the progression of the disease. You should now have a feeling for what things people with HIV or AIDS are more prone to. Like we've talked about PCP, pneumocystic pneumonia. We've talked about Candida. We've talked about a variety of infections, toxoplasmosis, several that they're more susceptible to. And then what else do you think is really important to know about HIV? Yeah, so the viral life cycle. And in general for this exam, since at the start we did a little review of viruses, right? So you should be familiar with some general things about viruses. So DNA viruses bring along what important enzymes with them? DNA polymerase, right? RNA viruses bring along RNA polymerase. Retroviruses have reverse transcriptase. And then what's key about plus sense RNA viruses versus minus or negative sense or antisense? What's the difference between plus or sense and minus and antisense? The plus sense can be used directly as a messenger RNA. So there's some multiple choice questions which go over kind of those general characteristics that we've talked about. So for HIV, you're gonna see some questions about the drugs, right? What's the important one that we've talked about now a couple of times? AZT, understanding AZT mechanism, that's very important. All right, so HSV 1 and 2. So we have cold sores and genital herpes. Now, what's something that's important to remember about HSV 1 and 2? Like a little factoid that we learned about. How about latency? Okay. So what's different between them in terms of latency? Where they are, right? So HSV1, where is it latent? Trigeminal nerve, right? And HSV2? Sacral, yeah. You should also recognize that with these HHV, HSV, these human herpes viruses, remember we talked about the fact that they can attack a variety of cells, right? Some B cells, some will be latent and neuronal cells, right? So not just HSV 1 and 2, but also HSV 3, right? Because that's how you get shingles. It's latent in the neurons. So with chickenpox, what does chickenpox look like? Little pustules, right? So you have these kind of blistery sores. Can you get chickenpox if you have never had it and then are exposed to somebody who has shingles? Yes. Yes, you can. So people with shingles are basically highly contagious. All right, now with HSV4, we talked about quite a few things. So HSV4, Epstein-Barr, EBV, what is the most common disease that people have when they have an EBV infection? Mononucleosis, right? But there are 3 other diseases that we talked about. What are the 3? Burkitt's lymphoma, which you see associated with what other 2 diseases? Easy one. So people with Epstein-Barr, HIV, and also Epstein-Barr and what? What's another big one up there, diseases? Malaria. Okay, so we have mononucleosis, Burkitt's lymphoma. The third one is nasopharyngeal carcinoma. So is Epstein-Barr EBV, is it an oncogenic virus? Yes. And then what's the 4th one? What else can it cause? Besides cancer, what about effects in the liver? Hepatitis, right? So we talked about hepatitis A, B, C, D, E, right? But we also talked about Epstein-Barr and cytomegalo also causing hepatitis. Okay, so let's move on to cytomegalo. Cytomegalo for most people, is it complicated or uncomplicated? Uncomplicated, right? Most people are asymptomatic. What population do we worry about with cytomegalovirus? Pregnancy. Right, so when we talk about categorizing, one of the things that you can see with cytomegalo, so you get abnormal growth, lots of defects, congenital defects. Microcephaly is one of them, and we also talked about another virus that causes microcephaly. What's that one? Zika. Okay, so then our last one in that family, remember these are all double-stranded DNA viruses, right? So they can be treated with what drug that we just talked about today? Acyclovir, right? Or valacyclovir. So HHV8 is Kaposi's sarcoma, and the population prone to that is what? HIV/AIDS. Okay, so then we have hepatitis A, B, C, D, and E. Which one is highest of concern? C. Why? Because there's no vaccine. That's right. Hepatitis A is not problematic, remember, and doesn't typically cause chronic liver problems. B and C do, C being the most severe with chronic liver disease, being oncogenic, right? So causing liver cancer and being the leading cause of liver transplants. So what's special about D and E? They're like satellite viruses, right? So D you only see D as a co-infection with B. So by getting what? What gives you protection against hepatitis D? The hepatitis B vaccine, that's right. And what about hepatitis E? Hepatitis A. And hepatitis E is really only a concern for pregnant women. For some reason it has, targets pregnant women and has higher mortality rates, bigger, more dramatic effects. So for the hepatitis, go through A, B, C, D, and E and look at the modes of transmission. Okay, so write a little note about transmission. Okay, so then we've talked a lot about influenza now. What do you need to know about influenza? Right, it's an enveloped virus and it has HA and NA spikes, hemagglutinin and neuraminidase. And what's special about hemagglutinin and neuraminidase? Right, attachment is which one? HA, right? And then release is neuraminidase. And we saw a little video today, that's because it helps with the cleavage, right? So that when they bud, they can be released. So important things about influenza. You should be familiar with antigenic drift and antigenic shift. What is drift? It's small point mutations, right? Can even be just a single, like I said, point mutation. It's changes in the sequence for the HA or NA spikes. So how does that compare with shift? That's drift. What's shift? Right, when you have a cell that's been infected with two different strains, right? And you have mixing of the genome. So you should be familiar with shift and drift. And then what drug did we talk about? Tamiflu, and how does Tamiflu work? It's a neuraminidase inhibitor. So it is not a cure, all it does is lessen the course of the disease. All right, so then we have our measles, mumps, rubella. How do we prevent measles, mumps, or rubella? Vaccine, right? MMR vaccine. Now, which two is rash the important sign that you would see? Measles and rubella, right? Mumps, what did we talk about with mumps? Right, swollen, right here, the swelling, remember the picture of the guy? And then also, for mumps, what do we worry about? I'll give you a hint. What do we worry about for male patients? Sterility, right? Because of the effects in the testes. So we worry about sterility. What about rubella? What's the major concern there? Pregnancy concern. Okay. Congenital rubella syndrome. So congenital effects or fetal death. Now for measles, we talked about one key thing that can help with diagnosis, something you could observe. What should you associate with measles? Before the onset of the full body rash, what can you see? Inside the mouth, Koplik spots. So you should associate those two things. Koplik spots, influenza. Measles has been in the news a lot. They've been talking about it a lot yesterday and today because measles has been on the rise. They've had a huge increase in measles-related deaths. One of the biggest concerns with measles, of course, is hearing loss. And the measles-related deaths have been due to unvaccinated people, younger children that are not vaccinated. And remember, we talked about the fact that you don't get the MMR, the first dose of MMR, until 1. But those that are unvaccinated are at a high risk, and there's been a dip in vaccine compliance, and so a big increase in measles-related deaths. All right, so HPV. So HPV, remember we talked about there being over 100 different strains, right? But which two are we concerned with most? (16) and 18. Because why? Because they're oncogenic, that's right. So, cervical cancer, 70 to 80% of the cases of cervical cancer are caused by HPV 16, but there are also pharyngeal cancers, throat cancers that are a result of HPV infection. All right, so let's move on to polio. Now, when we talked about like chicken pox, right, that's respiratory route of transmission. Many of these examples here, especially of these common childhood illnesses, are respiratory route. Right? What's the route of transmission for polio? Yeah, it's fecal oral, right? It is an enterovirus. So the enterovirus that they've been talking about in the news, the D68 that causes, that's been causing that polio-like paralysis, it's in the same family. So these have their effects typically in the summer months, and then you might see the cases of paralysis increase in the fall. So, and it's a fecal oral route of transmission, not respiratory route. So like your typical flu and cold, those respiratory routes, when people are cooped up, when it's cold outside and people are cooped up and have more contact with each other that way, that's when you see those, right? In the warmer months, you see more of the fecal oral route of transmission. So, polio, how do we prevent it? Vaccines, right? And there's two forms of vaccines. The oral vaccine and then the injectable vaccine. right? The inactivated. So the oral is a live attenuated virus, and the injectable is an inactivated virus. It's treated with formaldehyde, or was originally. So it's inactivated. Which one do we worry about reversion? The oral one, right? So that's the problem with any live attenuated viral vaccine, is that there's the potential for reversion. So back mutations. Okay, so moving on to Ebola. What do we need to remember about Ebola? It's hemorrhagic, right? So that kind of makes it a little bit different from the other ones that we've talked about. What about the route of transmission? Yeah, it's direct contact, right? And yeah, bodily fluids. What else about Ebola in comparison to some of these others, like chicken pox or influenza or measles? What's unique about Ebola? Very high mortality rate, right? But we talked about the exceptions, how it can vary, but typically a high mortality rate. All right, so our last virus is Zika. So, what do we need to know about Zika? It is mosquitoes. Which mosquito? Aedes. That's right. The Aedes mosquito. What's the other mosquito you need to remember? Anopheles, and that one is malaria, right? So the Aedes mosquito, Zika is transmitted through this vector, but how else? Yeah, transfusions, that's possible. And how else? Yep, mother to child, and how else? Sexual transmission. Right. So lots of modes of transmission. So what do you associate with Zika? What signs, symptoms? Yeah, so fever, so most people are going to be asymptomatic, right? Some people can get conjunctivitis and have a rash, fever, so have a short illness. What's the other thing that could happen, almost like secondary to Zika infection? Guillain-Barré. Yeah, Guillain-Barré. So GBS. So we talked about GBS when we talked about C. jejuni, but GBS is also an effect of Zika. So remember, Zika has a low mortality rate, but microcephaly, it is a pregnancy concern. So if we make a little list over in the right-hand side, pregnancy concerns. When you look at this list, which ones would you put on your list of pregnancy concerns, starting up at the top? HIV, then what? Yeah. How about HSV? HSV2? Then cytomegalo. Of course, hepatitis would be on there as well. What else? Rubella. And then Zika. Those would be the main ones that you should be able to pull out for pregnancy concerns. All All right, what about oncogenic viruses? If we make a list of yes,
The HB tested for certain now.
probably less likely, and probably less studied. So, pregnancy concerns, now we need to move on to oncogenic viruses. So starting at the top of our list again, which ones would you put for oncogenic viruses? Epstein-Barr, right? EBV, or HSV4, HHV4, right? Epstein-Barr. What else? Hepatitis C. HPV. And what's the other really obvious one? HHV8. Kaposi's. All right, so that takes us through really the kind of guts of the viral stuff. General background, key things associated with each viral disease, a little bit about drugs, categorizing them, right? How do you answer a multiple choice question? All right, so then we have just the two prion diseases. What's key about all prion diseases that we've talked about a lot? Yeah, they can cause death, right? And you can't do anything about it. They're not treatable. But what about what's infectious? Protein, right? This is protein, it's not nucleic acid. So of these, we have Creutzfeldt-Jakob disease, CJD, and Kuru. Which one is associated with cannibalism? Kuru, right? And its name comes from kind of like the delirium that people get. And then CJD, you get a form of dementia, right, that develops in death within a year. Now for CJD, we talked about the different types of CJD. So what are the types? Sporadic, familial, iatrogenic, right, and that's through medical procedures, right, and then variant, CJD, which we think people acquire from consuming, it's like consuming contaminated meat. All right, so you guys ready to flip the page?
Heck yeah.
All right, back side. Okay, so one thing that you should do, I have given you the genus or the genus and species, so you need to know those. And then you need to know the diseases. So, blastomyces dermatitis causes what? So it's in the fungi. So what do we call fungal diseases? Mycoses. Okay, so if it's caused by blastomyces, what do you think the disease is? It is blastomycosis, right? What about coccidioides? It is coccidioidomycosis. Okay, what about cryptococcus? This is a tricky one. It's cryptococcosis. Now, put a little asterisk there. Cryptococcus neoformans is a fungi and causes cryptococcus. Which one is that one easily confused with? Just name recognition. Look at your list. Cryptosporidium, which is a plasmodium. Okay, so make sure that you know those guys well. All right, then we have histoplasma. That causes what? Histoplasmosis. All right, then we have the tinea. So aspergillus causes what? Aspergillosis. Candida causes candidiasis. And then we have the Microsporidium and pneumocystis. Pneumocystis causes what? Pneumocystis pneumonia, or PCP. All right, so fungi. Now, if you look at the top of the list above tinnias, right? All of those guys, if you were going to categorize them, how would you categorize them? What type of mycoses? Systemic mycoses. And a lot of these guys then are going to have what characteristic? Dimorphism, right? And so in the human body, they're going to exist in a what form? Yeast-like form. So for systemic mycoses, how do you acquire them? What's the route of transmission? Inhalation. That's right. So we've talked about spores. What are fungal spores used for? Sexual reproduction, what else? Asexual reproduction. And because we have sexual reproduction and asexual reproduction, when we're talking about scientists or researchers or providers, what are they good for, the spores? What could you use spore properties for? What do you use like biochemical properties for bacteria? Identification, that's right. So fungal spores are really good for identification. Okay, so below the tinnias, we have aspergillosis, candidiasis, microsporidia, and PCP. What kind of diseases are those? What kind of mycoses? Opportunistic. All right, so let's go through them kind of one by one and pull out the key things, some of the key things that we talked about. The questions on these are pretty easy. You won't have any short answer on these. These are all multiple choice. So in terms of organisms or pathogens and short answer, the short answer are going to be relegated to the gram-positives. And remember, you're going to see some, like you saw before, only your list is going to be a list of a few gram-positive organisms, right? But the same thing, virulence factor, how it contributes to disease. So for these guys, the fungi and the protozoans, you're going to see multiple choice questions. So we've already categorized these first four: blastomyces, the coccidioidomycoses, cryptococcosis, histoplasmosis. Those guys are systemic mycoses. So, and we know you get them through inhalation. So blastomyces, what kinds of signs, symptoms, things? What should jump out at you in a question format that you should say, okay, that's blastomyces? What's this, where are the spores? How would you inhale them? Dust, right? And then this one can spread. Where can it spread? So you won't only see, yes, skin. So not just effects in the lungs, so pulmonary, yes, but it can become systemic. It can spread to the skin and cause ulcers. So those are the things that you should associate with the blastomycosis. Now, what about coccidioidomycosis? It's the same, right? Inhaling dust. But remember with these two, we talked about different regions, right? So the blastomycosis more wet, coccidioides more dry, desert areas. So then we have cryptococcosis and histoplasmosis, and they too have similarities. What should you associate with those two? Birds, right? Of some sort. Cryptococcosis is associated with what specific bird, though? Pigeons, right? And the spores are in the pigeon droppings. How is that different than histoplasmosis? That's right. So it's the droppings, the bird droppings or bat droppings, the bat guano makes a good fertilizer for the soil. So soil contaminated with bird droppings is a good source. That's then the reservoir. So the birds themselves are not, that makes sense, a reservoir. It's the soil contaminated with the bird droppings. Yes? Mhm. All right, so that'll get you through kind of the main high points of those four. Now, for the tinnias, if it affects the scalp, what do we call it? Tinea capitis. Feet, what's athlete's foot? Tinea pedis. Jock itch. Cruris. So then we have the nails, right? Unguium. So make sure that you can correctly match up tinea pedis, tinea unguium, tinea capitis, tinea cruris, right, with their location on the body. Okay, so then we have these last four are opportunistic mycoses. Aspergillosis. What did we talk about aspergillosis? Yeah, it's very invasive, so you should assist that with the lungs fill with mycelium. So the fungi just grows and fills up the lungs. What population is more prone? HIV immunocompromised individuals. And so this is a pulmonary disease. But also remember, I talked about it being a carcinogen, very carcinogenic. All right, candidiasis. So that can affect all parts of the body, right? We talked about diaper rash. What do you call it if it's in the mouth? Thrush, right? Important term to remember. So oral candidiasis is thrush. All right, so we have two more. Both of these, the microsporidia and the PCP, those affect what population? HIV/AIDS. What's unique about the microsporidia that can be used for diagnostic purposes? Can be directly observed. The polar tubule. Remember, that's the guy that sends out the polar tubule, and then all of the cytoplasmic contents go into the host cell. And then for our very last one, what disease should you associate with pneumocystis? Pneumonia. so now we're gonna move on to the protozoal diseases. Now, with these, you should be able to pull out those that have an important vector, right? So give me some examples of some arthropod vectors for these protozoal diseases. The tsetse fly, sand fly, right? Mosquito, kissing bug, right? That tritone bug, the kissing bug. And then besides vector transmission, what's the other important route of transmission? Ingestion. Yeah. So contaminated water or contaminated food. So when we look at this list, let's start with ingestion. Those are easy to pull out. Which ones would you get through ingestion? Giardia, what else? How about entamoeba? The crypto, right? Cryptosporidium, Cyclospora. So, when we're talking about Entamoeba histolytica, Cryptosporidium, Cyclospora, Giardia, what's going to be the disease? What is a patient going to experience? Diarrhea, right? So yeah, GI effects. Okay, so we also have one that can be fecal-oral route, can be hand-mouth, can be also sexually transmitted. What's that one? Toxoplasmosis. And then we have one other guy that's sexually transmitted. Trichomoniasis. That's right. So Trichomonas vaginalis causes trichomoniasis. All right, so then our trypanosomes. This is the African sleeping sickness and the Chagas disease. Leishmaniasis, right? Those are, and Plasmodium. Those are our important vector-borne illnesses. Now, a lot of these have reservoirs, warm-blooded animal reservoirs. But for example, Toxoplasma, what's an important reservoir? Cats. All right, tsetse fly, which one is that? That's right, African sleeping sickness. Kissing bug is what? Chagas disease. And that leaves us with leishmaniasis, which is what? Sand fly. Okay, and our Anopheles mosquito is malaria, which is what? What organism? Plasmodium. All right, so we had a quiz question on this today. What's important about trypanosomes? What's medically important, of world health importance? Right. No vaccine because of antigenic variation. So when we talked about protists, we also talked about motility, right? So flagellated, so flagellar motility versus amoeboid motility versus cyst. So trypanosomes are what? They're flagellated. And leishmania is also in that family of trypanosomes. It's flagellated. So we have African sleeping sickness has neurological effects, Chagas disease, right? You get those sores that can heal. Leishmaniasis, what type of leishmaniasis did we talk about? Cutaneous, right? So on the skin. Peter, you had a question? No, trichomoniasis and toxoplasmosis. Okay, so Entamoeba histolytica, how does it move? Amoeboid movement, but you acquire it because you acquire cysts, that's right, so you're thinking about cysts, and you have diarrhea. Those are kind of the high points, right, of Entamoeba histolytica. Now, what about Acanthamoeba? We talked about that one. In a multiple choice question, what kind of key terms, or what do you think you'd be asked about? What did we talk about with Acanthamoeba? That's right. Contact lens solution, no homemade saline solution. Don't put them in water in a shot glass, husband. Don't do it. Okay, so water contacts, so keratitis, we talked about that. Now, malaria. Malaria is really complex. We talked about a lot, and we're going to review it some next week as well because we're going to talk about treatment for malaria. So we'll review the life cycle. But remember, also with these guys, not only did we talk about movement, but we also talked about forms. So what is the infectious form? What do the mosquitoes actually inject? What form of the plasmodium? Well, I gotcha. You're stumped. The sporozoites. Then where do they go? Right, they go to the liver. And then they turn into what? Merozoites. And they're merozoites in the blood. And remember, we talked about the ring structure. So in the liver and the blood, what form? Merozoites. Infectious form that's passed from the mosquito? Sporozoites. Right, and remember with malaria, you're going to have fevers, and you're going to have this kind of undulating, the ebb and flow of fever. Fever comes, fever goes, and remember that that goes along with the life cycle. And you should be connecting, malaria with Burkitt's lymphoma, for example. So kind of connecting some of these things together. All right, so Cryptosporidium, Cyclospora, Giardia, we talked about those three all as being ingestion, diarrheal diseases. What's the difference between Giardia and then Cryptosporidium, and Cyclospora? Giardia has what type of motility? It is flagellated. Remember, it's a little alien, guys, with the two eye spots. And the flagella, right? The other two are then what? You ingest what? Cysts. So with our Cryptosporidium and our Cyclospora, we're talking about HIV/AIDS patients. All of those guys you can get through contaminated food and water. So we talked about cats and toxoplasmosis, but what else did we talk about with Giardia? What can give you giardiasis? Contaminated water and your dog or your cat, right? So there's lots of reservoirs for that one. And we talked about like daycare centers. So Giardia is a problem because it can prevent nutrient absorption. So it can cause malnutrition. All right, so now we've kind of covered these last two, toxoplasmosis, we've said fecal oral, we've talked about cats, You can also get it from undercooked meat, right? Most people will have toxoplasmosis and be completely asymptomatic. A lot of people have antibodies to it. But what population are we worried about with toxoplasmosis? Pregnant women and also HIV/AIDS patients. And this won't be on the test, but remember there's also that link with schizophrenia and homeless populations. Lots of interesting thing with toxoplasmosis. They're doing a lot of research on it now. And then trichomoniasis, we said is sexually transmitted. Okay, which sex is it asymptomatic, men or women? Men, that's right. And in women, in some cases, but it could be as low as like 30%. But what could you observe in an exam? Strawberry cervix. Okay, so that's about it. So now you have one sheet of paper, right? With some notes on it. This is your one sheet of paper. Don't make more paper. You only need this paper. All right, so we've got a little bit left. So next week, I don't know if you guys saw the announcement, but if you haven't done and don't have time to do the Connect assignments for this week that are due on Sunday or next week, you don't have to do them. You can do them as bonus. So if you missed assignments earlier, did that make sense to everybody how that was going to work? You can do it or not do it. If you don't do it, it doesn't matter. If you do it, those points will be tacked on, but the total point value will just be lower. It will be the total points for weeks 1 through 13. Okay? So anything you do for week 14 or 15 will be added on, and then all of that will still be the 10% of your grade. So if you've missed some points, you can recoup them. Yes? No, you could just pick some and not do the quizzes if you want to. That's what I would do. You think they are? Ah. Okay, do the quizzes. Yeah. But you learned so much. Look, okay, my oldest daughter is in what they call integrated math. That basically means math self-teaching. That's what I've discovered. So basically they have all these programs that they have to do and reach these goals every week. It's a lot like the Connect, only this is what you should be grateful for. Okay, if she misses a question, she has to do 5 more questions. So, think about it. Yeah. So the more anxious you get as the deadline is approaching, the more likely you are to miss a question, right? So if you miss a question and you have 5 more questions to do on top of whatever, 2 questions is 10 more, 3 more is 15 more, her head starts to go, yeah, Sunday night tears. You know, you gotta help me with this, I can't get it wrong because then I'll have to do 5 more. So, I feel your pain because I'm like, oh man. But it could be worse. That's my point. All right, so let's just look and talk a little bit about the list of gram-positives since we have some time. So next week we're going to compare and contrast again botulism and tetanus. That seems like a really long time ago, right, that we talked about this. We're going to compare them again, but we're going to go into more detail. I updated the slides. So what you see is probably what we're going to cover. I know Samantha's hoping for a snow day on Monday. She's already let it be known. I think even if this happens, we're going to get through that content. It's like 50 slides, right? Next week, one way or another. So We're probably not gonna have anything more than that. I'm hoping we don't have a snow day on Monday, so we can do that content Monday and Wednesday, and then we will review again for the exam on Friday. So don't hope for a snow day, because then it takes away our review day if we have to cover content on this day. So we have that. There's a little bit, just cool applications of micro, there's bioremediation. What else did I put in there? Oh, yeah, so tetanus, botulism, Botox, that's all kind of grouped together. Bioremediation, that's a short little blurb. We're gonna talk about the BT toxin. So, oh, and biofuels. So we're gonna compare ethanol and hydrogen. Okay, those two biofuels. So it's just a little bit. That's like a 15-minute discussion for each one of those things. All of those make for really good short answer questions. So it will be short answer questions over most of that stuff that we're doing next week. You guys usually are well prepared and do well on the short answer questions. So you're not gonna see most of the multiple choice content. In other words, we have already covered at this point. So the short answers will be about that stuff, in addition to the other few things that we've talked about, like the CRISPR. So there will be a few fill in the blank on this question, on this exam. Major terms. We'll cover some of that stuff again, hopefully on Friday. So, just to kind of look at the gram-positive organisms. So, for Clostridium, just kind of like a brief overview. So, Clostridium, what are the 4 main diseases? So I gave you 2, those are easy ones. What are those 2? Tetanus and botulism. All right, so for tetanus and botulism, what's important to remember to differentiate them? Right, rigid paralysis, flaccid paralysis. Flaccid paralysis is botulism, right? That's why I need Botox here so I can't frown all the time. I keep telling my husband, I'm like, if they would just poke me right here, then I couldn't frown all the time, and they could poke him in the feet, and his feet wouldn't sweat all the time. You know, maybe we could get a twofer, I don't know. But, so we're gonna talk about some uses of Botox, besides just keeping me from frowning. Just seriously, just right here, then I couldn't frown. So besides those 2, what are the other 2 for Clostridium? Gas gangrene, which is what? What it is. It's basically, yeah, it's gangrene, right? So that is C. perfringens. And then what? Diarrheal disease. So it is what? C. diff. All right, so for Clostridium, what do you think about virulence factors? What's the major virulence factor or category of virulence factor that those guys all have? Exotoxins. That's right. So all 4 toxins. All right, what else did we cover? So we did Clostridium. Oh, we talked about Bacillus. So we talked about what? What should you remember? What's the big one? Bacillus what? Anthracis, right? Which is anthrax. Now, we talked about some key things. So with anthrax, what about the route of transmission? There are multiple ones. There's 3, that's right. What are the 3? Inhalation, right, in through the wounds, and then ingestion. So we've made cards before. You guys should go through and put the genus and the species, because remember, for Clostridium, we have Clostridium botulinum, we have Clostridium tetani, we have Clostridium difficile, we have Clostridium perfringens, right? So you need to put the genus and the species with the disease, important virulence factors, short answer question, right? Okay, so we talked about Bacillus anthracis, and then we also talked about one that can cause foodborne illness. Which one is that? Be serious. Yes. That's right. Be serious. I had terrible diarrhea. All right, then we talked about staph, right? So for staph, what are the two species? Staph aureus, Staph epidermidis. Which one is pathogenic? Staph aureus. Now, if you would choose Staph aureus on a short answer question, what would you write about for virulence factor? Yeah, you could write about anything. Coagulase, chemolysins, right? Uranidase, but what would be a really easy one? Blank, blank, Staphylococcus aureus. Methicillin resistance, right? Antibiotic resistance, methicillin resistance, vancomycin resistance, MRSA and VRSA. All right, what about diseases associated with Staphylococcus aureus? Red, peeling skin. Scalded skin, right? The SSSS, right? What else? What other disease? High fever. Red skin. Toxic shock. Right. What about super antigens and staph? What else can you get? What other kinds of diseases associated with staphylococcal enterotoxin? It's an enterotoxin. Yeah, intestines, right? Foodborne illness. All right, we also talked about lactobacillus, right? You guys should be pretty familiar with lactobacillus, normal flora. Right? Okay, streptococcus. So we talked about all kinds. Streptococcus, what you find in the mouth, and it's associated with dental cavities. Streptococcus mutans. What about scarlet fever? Pyogenes, Streptococcus pyogenes that's been lysogenized causes scarlet fever, right? What else do you worry about? So Streptococcus pyogenes causes what common illness? Strep throat. Strep throat left untreated can then turn into what? You get nodules by the elbow, cardiac effects, lifelong cardiac effects. Rheumatic fever. What about neonatal sepsis? S. agalactiae. So, S. agalactiae is what type of strep? Group what? It is. It's a GBS, Group B strep. And then what about pneumonia? Which one? S. pneumoniae, right? Streptococcus pneumoniae. Okay, so that leaves us with 3 main genera left. Listeriosis is caused by what? Listeria monocytogenes. What's the route of transmission? Fecal oral, right? Ingestion. Population we're concerned about? Pregnant women, right? Can cause fetal death. And then our last true gram-positive is Corynebacterium. What does Corynebacterium cause? Diphtheria, that's right. Diphtheria, respiratory illness, right? What's the one thing, it can also be cutaneous, you can have wound diphtheria. What is the key thing about diphtheria that you can see that can be useful for diagnosis? The gray membrane. So if it's an ulcer on the skin, a gray membrane, or that gray fibrous membranous stuff that has like white cells in it and stuff that patients can cough up, right? The pseudomembrane. Diphtheria, what kind of virulence factor? It's an exotoxin. With Listeria, what did we talk about? That's right. They can, and they can grow in what type of cells in the body? Phagocytic cells. So they reproduce in phagocytic cells. That's an important virulence factor. Listeria, yeah. And then we have tuberculosis and leprosy. What's the genus? Mycobacterium, right? So M. tuberculosis and bovis can also cause a small percentage of tuberculosis cases. And then we have M. leprae, which causes leprosy. So tuberculosis is respiratory illness, right? So what are some terms you should associate with tuberculosis? Yeah, tubercle, right? What happens to the tubercle that forms? So first of all, what's happening there in the lungs? What kinds of cells are there in the lungs? Immune cells. The macrophages, right? So the bacilli can invade the macrophages, reproduce there. You get those tubercles that form, and then they can take on a cheese-like consistency. What do we call those? What type of lesions? Caseous lesions, cheese lesions, right? And then they can harden, calcify into what? A gone complex. So transmission, respiratory route of transmission can also be GI transmission. What about leprosy? What's the difference with leprosy? What's the route of transmission? Direct, prolonged contact. So there's a neuronal form, right, and the progressive form, where you see the characteristic disfiguring nodules that form. All right, so that should get you started. Now for the Mycobacterium, what would be, for either M. tuberculosis or M. leprae, what would be an important virulence factor? It's in the name. Mycolic acid. That's right. because the mycolic acid makes them really resistant, right? Resistant to antibiotics, resistant to phagocytosis, makes them very, very resistant. All right. So, review the drugs. We'll finish up next week. Friday, we'll try to squeeze a review in class. And then I will also give you the list of short answer questions next week. Okay? All right. Have a good weekend, guys. I'll see you on Monday, even if Samantha's not here. I'll see the rest of you.